Good.
Well, and so I'm going to just introduce you, Luke, briefly, but just to remind everybody, this is our weekly seminar, and it's also a class for some students. We'll have people trickle in because in addition to students, we have our own faculty. We have faculty potentially from other departments. We have FDA colleagues that may join us. And so this is kind of the hour where people are switching in classes, having lunch hour. So anyway, so don't worry too much. People will be coming and they may have to exit a little bit early, but we do have an hour to hear your story, Luke. And so just for everybody out there, Luke is Dr. Howard, I should say, is a longtime friend, and we've met over polyphenols and berries for sure. He is a graduate of Purdue, but well, I should say his undergrad was at Purdue, and his graduate work was at Arkansas, where he also spent the bulk of his career. And if you were on just a few minutes ago, you will hear that this is one of his last acts as an active faculty member at Arkansas. He's going to retire, but we're hoping he does not give up any of his research work. Maybe he's just going to transfer up north a little bit in Indiana. I'm assuming that's north, right? My gosh, I should know my
Yeah, it is from here for sure.
Right. So anyway, phenomenal scientist, and I'm really pleased to introduce Dr. Howard for our Thursday seminar. And so with that, please, the floor is yours.
Hey, thank you, Dr. Burton Freeman, for that wonderful introduction. I'm very pleased to be with you here today. I was planned to give this presentation at the Berry Health Benefits Symposium earlier this month in Florida, and we got postponed, which was a travesty. I could have escaped all this nasty weather we're experiencing now, but I'm certainly pleased to share this information with you all today. So the title of the talk is Anti-Inflammatory Properties of Blackberry Phenolic and Volatile Extracts. Okay, and now I can't advance the slide. It's always something. There we go. So we all know about health benefits associated with consumption of fruits and vegetables. And this is because fruits and vegetables contain a wide array of phenolic compounds. They're also an excellent source of dietary fibers, vitamins. They contain low levels of carotenoids. The berries do anyway, but certainly vegetables are a rich source of carotenoids. So all of these fruits and vegetables are rich in phenolic compounds that we all know provide antioxidant and anti-inflammatory properties and are thought to protect against a myriad of chronic diseases. So we're primarily interested in protection against inflammation, which is the body's response to harmful stimuli such as microorganisms or an injury. Also, the Western diet can put us in a pro-inflammatory state. So a diet high in carbohydrates and fat. We're in a chronic pro-oxidative and inflammatory state. Dr. Burton Freeman's certainly done a lot of great work in this area. So we need to consume a diet that's rich in antioxidants and anti-inflammatory agents. So after the stimulus of inflammation to a harmful stimuli, the mechanism starts with intervention of immune cells such as neutrophils, monocytes, macrophages, and mast cells around the site of inflammation. And then these immune cells release inflammatory cytokines such as TNF-α, IL-6, and IL-10. And then these cytokines will release inflammatory markers such as nitric oxide, prostaglandins, and these markers then create an amplified inflammatory situation. So nuclear factor kappa B is able to regulate the expression of cytokines, and if these are overexpressed or if the inflammation does not resolve, there's a situation of chronic inflammation that contributes to the initiation and promotion of chronic diseases. As you can see here, cancer, asthma, cardiovascular disease, metabolic syndrome, neurological diseases, disorder. So we definitely need to control inflammation as well as oxidative stress in the body. So we all know about phytochemicals. These are the natural compounds found in fruits and vegetables that afford protection against chronic diseases. So these are other than traditional nutrients such as proteins, lipids, carbohydrates, and vitamins and minerals. There's thousands of different phytochemicals found in plants, but most of the work, including work from Dr. Burton Freeman and myself and many others, we focused on the phenolic compounds because these are very potent antioxidants and anti-inflammatory agents, especially the anthocyanin pigments. But a lot of work's also been done on the tannins, the proanthocyanidins, and the lagitannins, and some work on flavonols such as quercetin derivatives. So today, what I'm going to talk about are essential oils that fall under the classification of terpenoids, which is a broad umbrella that also includes carotenoids and steroids. So why focus on essential oils? Well, if you look at the essential oil literature, it's been reported that these volatile compounds, have very potent anti-inflammatory activity. So that's kind of what led us to start looking at the volatile compounds in berries. Previously, we knew that these compounds are important for the aroma of berries, but we really didn't know if they played a role in health promotion. So first of all, the phenolic compounds are very good antioxidants. Why is that the case? Any phenolic ring that contains hydroxyl groups can scavenge free radicals, which makes it a very effective antioxidant. So we start classifying the phenolics based on the number of phenolic rings. So we can have the simple phenolic acids, including a hydroxycinnamic acid, chlorogenic acid, which is a very high concentration in blueberries, although my favorite source is coffee. So plenty of chlorogenic acid in coffee as well. Then we get to the flavonoids with multiple aromatic rings and a heterocyclic middle ring here. But the anthocyanins have received the most attention. Of course, these are the compounds responsible for the pigmentation of berries. And this is why berries always rank high in terms of antioxidant capacity, is because they have very high levels of these anthocyanin pigments that almost always have a sugar attached here at carbon 3. We're also concerned with flavanols, the precursors to the tannins. So we're not seeing, well, I've got it blocked here, and then we're not seeing a proanthocyanidin, which are just polymers. But here's a trimeric procyanidin. So we just start forming oligomers and polymers of the flavanols to get the oligomeric and polymeric procyanidins. And then with other berries, including blackberries, we have lagitannins, okay, which break down to ellagic acid. And then, of course, when they're metabolized by the gut microbiota, these are broken down into simple phenolic acids, including urolithins. And there's also been a lot of work done on still beans, resveratrol in grapes, and pterostill bean in blueberries that have shown important health benefits. So what about volatile compounds? Volatile compounds are responsible for the aroma of plants, including berries. And the volatiles are odor active at very low concentrations, from parts per billion to low parts per million. They're volatile as a result of their low molecular weight and high vapor pressure. The higher the vapor pressure at a given temperature, the lower the boiling point. Hence, compounds with high vapor pressure are volatile. If blindfolded, one can easily identify a fruit by its distinctive aroma, as we can see in the top right here. Very easy to identify a strawberry, even if you're not looking at it. These volatile compounds are nonpolar, hence they're analyzed by gas chromatography. And we know that berry aroma is complex, so we typically see over 100 volatile compounds present in a GC chromatogram. And the aroma of fruit is typically due to a large number of volatiles, although a lot of the strong odors, the compounds may be present at a very low concentration, low parts per billion. So volatiles and aroma. How do humans perceive odors? This is through olfaction or chemoreception, forms the sense of smell. So olfaction occurs when the odorants bind to specific sites on the olfactory receptors located in the nasal cavity. These signals are transmitted to the olfactory bulb, and then sensory input will interact with parts of the brain responsible for smell identification. So each volatile has a unique aroma, but when mixed, a totally new aroma is perceived. So if you look at a lot of these volatile structures, here we see benzaldehyde, where you get an almond-like aroma. So here we're talking about an aldehyde, limonene, which gives us a citrus aroma, which is a monoterpene classification. Linalool, also a monoterpene, gives us flowery. Alpha terpineol, another monoterpene, gives us a lilac aroma. Hexanol is an alcohol, and this one gives us fatty or fruit or woody aroma. Ethyl butanoate gives us cognac. Beta ionone, which is a C13 norisoprenoid, gives us a violet aroma. So this compound is actually a breakdown product of beta carotene. And then we also can have a ketone, raspberry ketone, which gives us a sweet berry or floral aroma. And this compound has actually been touted for weight loss and fat burning. So if you look down here, we see the berries aroma. I also have cannabis. Why would I put cannabis here? Well, first of all, if you do a search on monoterpenes, the first thing that's going to pop up is cannabis. Cannabis is loaded with monoterpenes, as are hops used in brewing. So we actually analyze hops and cannabis in the lab. And a lot of the same monoterpenes that are in berries are also found in cannabis and hops, although we tend to see much higher levels of these volatile compounds in cannabis or hops. But the aroma is quite different because there's a different composition. So the biological significance of monoterpenes, as you can see here, they're used to attract insects, okay, for pollination. And they also can deter some animals or varmints because they do have a strong aroma. But these natural volatile extracts called essence are used in a number of different foods, drugs and nutraceuticals, food and beverages, cosmetics. So if on the label it says natural flavor, these are the natural volatiles that have been extracted from plants, concentrated, and then you can add them to all these different food products, including perfumes. And on the right, you actually see the blueberry essence. This is how we buy the commercial essence. That's the volatiles are concentrated about 150-fold. So we strip these compounds off when we concentrate juices. So as we evaporate the water, the volatiles are removed with the water and they're collected and sold as natural flavor. So the objectives of this study were firstly to isolate and characterize polyphenols and volatiles in 16 Blackberry genotypes. And then secondly, to compare antioxidant and anti-inflammatory activities of polyphenol and volatile fractions from 3 selected Blackberry genotypes. So the materials, the blackberries were grown at the University of Arkansas Fruit Breeding Station in Clarksville, Arkansas. So those of you that have never been to Arkansas, it's actually a beautiful state. If I was looking at this picture, I might think this is California, but we've got a lot of mountains, really large hills, but Ozark Mountains. So it's a beautiful state. So Pauline Murine, my master's student, did this work. So she harvested the fruit when fully ripe at the Fruit Research Station. And they were transported in coolers to the food science department and immediately analyzed for pH, titratable acidity, and soluble solids. So we use some advanced breeding selections. This is Dr. John Clark, who has the largest public blackberry breeding program in the world and has released many blackberry varieties that are grown worldwide. So for those of you that like blackberries, I definitely want you to try these sometime. This is one of his newer releases, Sweet Arc Ponca. These berries are incredibly sweet. One of the problems with blackberries, at least that I've experienced over the years, is you eat a couple that are super sweet and then you get a couple that are really sour or astringent. I don't know what he's done. It's like uniform ripening, but these berries, I ate a whole pint and about 95% of them were sweet. So this is a super blackberry that I think is going to do very well in the market. So there's a shot of the blackberry genotypes. We looked at 9 advanced breeding selections. So breeders are always selecting for new varieties that have improved horticultural traits, such as yield, color, size, shape, insect and disease resistance. And we also looked at 7 named or commercial cultivars have names, as you see here. So first thing we did was analyze the volatile compounds. So we use solid phase microextraction. So you grind up the berries and this spemy fiber goes into the headspace of the sample. You can probably see it better here. It's held in there. The volatiles will absorb onto the spemy fiber, and then it's retracted and injected into the gas chromatograph. So to do this, we used a temperature of 60 degrees C and absorb the volatiles on the spemy fiber, and then we inject them into the GC and get a number of compounds. Yep. So the first thing we analyze were soluble solids to titratable acidity ratio. So why do we do this? This is an important quality measurement for fruits. The higher the soluble solids to titratable acidity ratio, the sweeter the fruit and less sour. Previous work has shown that blackberries should have a soluble solids to titratable acidity ratio of 10 to 13%. So you can see that a good bit of the advanced selections and commercial varieties exceed 10% SS to TA ratio. And a couple were quite high, A2587 and advanced selection, as well as Primark Traveler, which indicates very sweet berries and possibly overripe berries. Okay, next we looked at blackberry anthocyanins. I love working with blackberries because they have a very simple anthocyanin profile. For those of you that have ever worked with blueberries, you'll see over 25 different anthocyanins. So blackberries are easy. There's 5 major anthocyanins, all cyanidin derivatives. And cyanidin 3-glucoside typically accounts for over 80% of the total anthocyanins in blackberries. But we also see the rutinoside, xyloside, and then a couple of acylated anthocyanins, malonyl glucoside, and the 3-dioxylyl glucoside. So we looked at the total anthocyanins as the sum of all of these individual anthocyanins, which we see here, the total anthocyanin content. So we see that these advanced selections and varieties vary 2.73-fold in total anthocyanins. So most of them are up around 150 or even higher. This A2587 in very high levels, almost 250 milligrams per 100 grams. So blackberries are comparable to blueberries in terms of total anthocyanin content. We also looked at the flavonols in the blackberry genotypes, and they're mostly quercetin derivatives. We also see a couple of ellagic acid conjugates, ellagic acid hexoside here, and a methyl ellagic acid pentaside, but most of these are quercetin derivatives. And the major ones are, you look at compounds 4 and 5, the quercetin glucoside, as well as the glucuronide, and this compound with a long name is also present at high concentrations. But the total flavonols, which we'll see here next, are about 10-fold lower than the levels of total anthocyanins. But again, this variety or advanced breeding line, A2528, looks very good in terms of total flavonol content. Next, we look at the total volatile content of the Blackberry genotypes, and we see a 2.3-fold variance in total volatiles. So while we were doing this, we wanted to pick 3 Blackberry genotypes that varied in volatile concentration to look at anti-inflammatory activity. So after getting these results, we selected A2587, which had the highest level, and then one with a medium level, Natchez, and then the lowest concentration, A2528. So our hypothesis was that the genotypes with the higher volatile content would have the greatest anti-inflammatory activity. So if we look at the volatiles, breaking them down by class, we see over 160 different compounds, but the aldehydes were the predominant class of volatiles, followed by esters, monoterpenes, alcohols, and then acids. And some of the major individual components were hexanal, an aldehyde, ethyl acetate, an ester, and linalool, a monoterpene. Now, flavor or aroma of fruits is very complex. And we found out when we analyzed these samples, you can't just look at the total volatiles. So this was a work done by Wang et al. In 2005, where they looked at the major odorants in Chickasaw blackberries. This is important because Chickasaw came out of Dr. Clark's breeding program, so it shares some of the same genetics of the genotypes that we investigated in this study. So they actually determined flavor dilution values for individual compounds. So the higher the value, okay, the more potent the odor. So this is where you dilute until you can't detect an odor. So for a couple of these, ethyl butanoate and ethyl 2-methylbutanoate, you had to dilute it 2,048 times before you couldn't detect it. Same way for geraniol. And the others are not quite as potent, but still pretty strong odorants. The thing that's interesting is besides linalool and geranirol and 2-hexenal, these other compounds are present at a very, very low concentration. So if we looked at the concentrations of these major odorants in the Blackberry genotypes, we see a much different picture than if we just rank them according to total volatiles. So A2547, which also was high in total volatiles, you can see here comes out #1 in major odorants, but then the sweet archponca comes in second, and this one was in the middle of the pack in terms of total volatiles, and then A2658T is third. So if you look at the correlation between total volatiles versus major odorants, it's only 0.36%. So this indicates that the measurement of the total Okay, so now we're ready to prepare these phenolic and volatile extracts to look at anti-inflammatory activity. Our goal was to obtain fractions of phenolics and volatiles with concentrations similar to those in fresh berries. So for the phenolic extract, we took 30 grams of berries and extracted with the methanol water formic acid, and then we evaporated and brought that back up in solution, ran it through a solid phase extraction with Setpack C18 cartridges, eluted the phenolics with 70% ethanol, evaporated that solvent, and brought it back to 30 mils. So we started with 30 grams of berries and we adjusted it to 30 mils with acidified water. For the volatile extract, we used 300 grams of berries plus 300 mils of water, plus a large concentration of sodium chloride. That was to inactivate the enzyme lipoxygenase. Of course, lipoxygenase will oxidize polyunsaturated fatty acids, so you'll get large levels of hexenal and hexenol if you don't inactivate lipoxygenase. And then we did a low temperature distillation, 40 degrees C, and we collected the first 200 mils of the distillate to get our volatile fraction. So we looked at the composition of the 3 selected blackberry varieties that varied in total volatiles. And as expected, they also varied in total phenolics as well as total anthocyanins. But the thing I want you to note here is look at the scale. Okay, total phenolics, micrograms per mil, total volatiles, nanograms per mil. So these are the fold differences we're looking at in terms of concentration between the volatile fraction and the phenolic fraction. Okay, the phenolics are higher, 15,000 times higher, almost 4,000 times higher, and 8,000 times higher than the volatiles in these fractions. We measured the antioxidant activity using DPPH assay, and this followed the same trend as total phenolics. So that's good for the phenolics, but the low levels of these volatiles, we could not detect any antioxidant activity due to the very low concentration. So we looked at the volatile composition of the 3 selected blackberry genotypes, and you can see they vary in composition. So A2587 is high in monoterpenes, alcohols, and sesquiterpenes, and Natchez is the next highest in the monoterpenes, alcohols, and sesquiterpenes, whereas A2528 has fairly similar levels of the major volatiles, the monoterpenes, esters, and alcohols. So another thing we looked at was testing a mixture of volatile standards. We were wanting to confirm that the anti-inflammatory effects were due to volatiles and not possibly due to other compounds co-extracted or distilled during the low temperature hydrodistillation treatment. So we prepared a mixture of these standard volatiles, okay, at concentrations that we had already found measuring the values in the blackberries. So you can see it was a whole host of volatile standards here. It came up to 407 nanograms per mil. So to examine the anti-inflammatory capacities, we used the RAW264.7 cells, okay, which are grown for 16 hours. So we looked at a preventive treatment with the volatile extracts that were diluted 2, 4, or 8-fold, and the phenolic extract that was diluted 10, 20, or 40-fold. So those were incubated for 1 hour. And then we challenged with the LPS for 24 hours. And then we measured nitric oxide, IL-6, and TNF-α. So first of all, we looked at cell viability. So none of the treatments reduced cell viability following 1, 2, or 3 hours, at least not significantly. However, some of the phenolic compounds stimulated cell viability. I'm not quite sure what to make of this, but in some papers, they talk about increased viability, maybe due to compounds interfering with energy metabolism, which can increase the MTT metabolism. It's also been reported that you can get chemical reduction of MTT by phenolics. But the good news is we didn't see any cell cytotoxicity with our 1-hour treatment. So let's look at the anti-inflammatory results. And this was exciting. First of all, looking at nitric oxide production. It's a lot of data here, so I've tried to have a little cheat sheet at the bottom. So we see a nice stimulation of NO production with the LPS challenge. So here, these are each varieties, 10, 20, and 40-fold. This is one variety, A2528. 3 dilutions of the phenolic, and then these are the volatiles, 3 dilutions, phenolic, volatiles, phenolic, volatiles. The volatile extracts worked much better than the phenolic extracts, as you can see here, here, and here, in inhibiting the secretion of nitric oxide. So all of them worked effectively, better than the positive control. With the phenolics at the 10x dilution, we see 24 to 42% inhibition of NO, and the volatiles at 2x dilution, 51 to 63% inhibition. In the standard volatile mix, we see comparable to the volatile fraction, 59% inhibition. So this is remarkable, again, considering the concentrations. So you can see here, phenolics 10x, that's 432 micrograms per mil, whereas the volatile at 2x is 142 nanograms per mil. So these volatiles are very potent anti-inflammatory agents. Here we see the results for IL-6. And again, we see a nice challenge with LPS, high level of IL-6, and then phenolics, volatiles, phenolics, volatiles, phenolics, volatiles actually behave quite similarly in terms of preventing the secretion of IL-6. So the phenolic extracts at 10x dilution, 35 to 53% inhibition, and we see a similar response with the volatile extracts at 2x dilution, 34 to 60% inhibition. However, the standard mixture of volatile standards was very effective at inhibiting the secretion of IL-6, 80% inhibition. Then TNF-α, see pretty much the same thing. Now again, the volatiles actually work better than the phenolic fractions. Okay, so the phenolic extracts at 10x dilution, 28 to 44% inhibition, but the volatile extracts 61 to 73% inhibition, and the standard mixture of volatiles also was very effective, 75% inhibition. So these results actually confirm a previous study that was published in Antioxidants by my colleague in food science, Dr. Sun-Ok Lee, where they looked at 6 different berries: blackberries, red raspberries, black raspberries, blueberries, strawberries, and cranberries. So all of these berry and volatile fractions showed the ability to prevent the secretion of NO, prostaglandin, COX-2, IL-6, and TNF-α. In this study, we used the 50x dilution. Higher dilutions were not active, okay? But in this study, they also showed that these volatile and phenolic fractions reduced the nuclear translocation of NF-kappa B by blocking phosphorylation of P65 and degradation of the inhibitor of kappa B. So what's going on? These inflammation pathways always give me a headache. I am not a nutritionist, but when we get a challenge from diverse stimuli such as LPS, okay, we stimulate the NF-kappa B pathway. So the NF-kappa B is naturally present, but is inhibited by the inhibitor of kappa B. So upon stimulus, these pattern recognition receptors activate the IKB kinase, IKK here, okay, which leads to degradation of the inhibitor of kappa B. So this occurs by polyubiquitination, binding of ubiquitin molecules to trigger the signal, leading to degradation of protein by proteasome, and also by phosphorylation of protein subunits such as P65. So these NF-kappa B's dimers enter the nucleus and initiate inflammatory gene transcription, leading to the production of these inflammatory markers, inflammatory cytokines and chemokines. Thus, by blocking phosphorylation of P65 and degradation of the inhibitor of kappa B, these berry volatiles suppress cytokine and chemokine expression, and hence can help suppress the inflammatory process. So what's known about metabolism of volatiles? We're starting to identify a lot of the metabolites, polyphenols. Unfortunately, there's very little information available on volatile metabolites. Okay. We do know that with monoterpenes, they're lipophilic, so we do get rapid dermal absorption, max plasma level in about 10 minutes. And we know that inhalation is also possible. So that's a big advantage over phenolics. Some studies report up to 54 to 76% pulmonary absorption is reported. So these compounds might play an important role in protecting against inflammation in the lung. The metabolic fate depends on the chemical structure. So there's evidence of both phase 1 and phase 2 metabolism. The majority of these compounds identified in the plasma are present as glucuronides. Okay, so they are undergoing phase 2 metabolism. And we also see excretion through the kidneys with the phase 2 metabolism, 60% or expired air as carbon dioxide. And we can get other changes too. Due to the P450 enzymes, we can get allylic oxidation, which changes the structure of these volatile compounds. And we can also get ring partial closure. So if the native compound is linalool, we can actually get partial ring closure and convert linalool to alpha terpineol, which can be converted to nerol and geraniol. But we do need a lot more information on the bioavailability of these volatile compounds, especially the monoterpenes, and determine if these metabolites reach the targeted organs. So conclusions, blackberries contain a complex mix of volatiles that impact both aroma and anti-inflammatory activity. Despite being present in the berries at much lower concentration than phenolics, volatiles show comparable or greater ability to inhibit secretion of inflammatory cytokines. We believe, therefore, that volatiles may play an important role in health promotion. The fact that the 3 genotypes varying in total phenolic or total volatile content all showed comparable ability to inhibit secretion of inflammatory cytokines may be good news, may mean that all blackberries work as anti-inflammatory agents due to the phenolic and volatile content, but it certainly shot down our hypothesis that the genotype with the highest levels of volatiles would have the highest anti-inflammatory activity. All 3 genotypes worked similarly if you look at the total of all 3 different anti-inflammatory markers. So we definitely need more work on bioavailability, ADME, absorption, distribution, metabolism, and excretion. We need to understand if there is synergy between these volatile compounds and other phytochemicals such as phenolics. However, it is going to be very challenging since these volatiles are present at such low concentration in the berry to be able to identify and quantify metabolites in plasma and urine is going to be very challenging. It's the same problem we have with the phenolic compounds. There's a lack of standards on volatile metabolites. Okay, now hopefully, and this be the last study I'm probably involved with at the University of Arkansas since I'm retiring. But we've actually started this project. We're looking at the anti-inflammatory effect of berry flavored water. You may have seen, this is one brand, Hint Water, on the market. So all this product has is natural berry flavor. Okay, so in our study, we're going to look at a berry mix with blueberry, blackberry, and cranberry. And the first part of the project, we determine the consumer rejection threshold for concentration of berry flavored water. Typically, with a natural essence used to flavor waters, it's concentrated 150-fold. So you dilute it 150-fold to get it back to the level in the fresh berries. And we found out from a rejection threshold consumer test that we could concentrate, only have to dilute 12-fold. So we're actually more than 10-fold concentrated than what would be in a commercial berry flavored water. So we're actually getting ready to start objective 3 here to determine the ability of consumer preferred berry flavored water to reduce acute inflammation resulting from exercise heat stress in human subjects. So in this study, the subjects will be lifting weights twice a day to induce inflammation, and half the subjects will consume the berry flavored water twice a day prior to lifting, and the others will just get regular water. And then the last objective, we'll look at the bioavailability and anti-inflammatory effects of volatiles in obese and normal human weight subjects following consumption of the consumer preferred berry flavored water. So we're going to collect blood after 1, 2, 3 hours after consumption and measure inflammatory cytokines, TNF-α, NO, IL-1 beta, IL-6, IL-10, and NF-kappa B. So hopefully we'll be able to show that the berry volatiles do protect against inflammation in human subjects. That would be cool. So lastly, I'd like to acknowledge my collaborators and students. So my collaborator in the Department of Food Science, Dr. Sun-Ok Lee, Cindy Brownmiller, my expert technician that helped the students with the GC and HPLC analyses. Katie Moore, who's now in dental school at the University of Iowa, started this work on cranberry volatiles, and she saw the same thing. The cranberry volatiles work just as well as the cranberry phenolics in terms of inhibiting the secretion of NO. Pauline Murine from France did this work as part of her master's thesis. And my other student that happens to be from France, Laura Lefebvre, is working on blueberries. And she's also looking at anti-inflammatory effects of blueberry volatiles, doing some nice cell culture work down at the Arkansas Children's Nutrition Center. So with that, I say thank you very much, and I'd be happy to entertain any questions you might have.
All right. Well, Dr. Howard, that was as phenomenal as expected. Thank you so much. This is really super exciting. I can't wait to talk more. But I know that there's questions out there, and I think Uday might have one because I can see her pop up on the list. Uday, do you have a question for Dr. Howard?
Oh yes, I have some questions. Howdy, Dr. Howard. I'm also doing some terpenoids analysis on herbs and spices. And yes. The major one I'm working with is a diterpene called carnosic acid. And yeah. I observed this during my extraction, which is a liquid extraction. There's a lot of degradation during my extraction. I'm wondering, in those terpenoids in berries, if there's also you observe some degradation during the extraction process?
Yeah, great question. Yeah, and it's not only degradation, since these compounds are volatile, and we've actually looked at this in the cell culture. You're incubating at 37 degrees C, right? Mm-hmm. So these volatile compounds, they're diffusing out. Yeah. So you've got degradation, you've got chemical degradation or oxidation. We've seen this too, and I don't understand. They sell the essence in plastic buckets. There's even degradation they should be stored in glass, which would be a better way to do it. But yeah, degradation is certainly an issue. But this is something we fight with, other phenolic compounds as well that are unstable over time. But no, it's certainly a concern. I don't know how long we can actually save these extracts, before they start losing bioactivity. So that's a great question. Still a lot of research to be done.
Yeah, and also, is this only monoterpene in berries? Because herbs and spice, I have looked, there's some diterpene and triterpene as well.
Right. And, we haven't looked at those. So you said you use a solvent, right, to extract your diterpenes. And there's like a lot of people are looking at triterpenes too in cranberries. So there typically you're using a solvent, and then you use those. Do you use HPLC to measure those?
Yeah, I use HPLC.
We're going more after the monoterpenes and the compounds that have a higher vapor pressure. And so we go use gas chromatography.
Yeah.
So the major monoterpenes, I mean, it varies according to berries. A lot of them have the same monoterpenes, but one might be real high in linalool and another one high in alpha terpineol. It's just all across the board in terms of the major individual compounds in each berry. That's why each berry has a different aroma.
Yeah, those terpene standards smells very strong. Like right now, I even wear masks in the lab. I can still smell it.
Yeah, absolutely. Well, it's a good smell. Anyway.
Yeah, it's pleasant. And one more question. During the volatile compounds extraction, you mentioned there's evaporation. I'm wondering how long you take during the evaporation process.
When we try to collect the volatiles to make our own essence for the extract, we're doing it about 30 minutes. And that's a good question, because I should have pointed that out. When we measure the total volatiles in the berries, or we just mix the berry into a puree and then we use the SPME, we see 10-fold higher concentrations doing that. We used a low temperature distillation of 40 degrees C. Okay, so we're only getting about 1/10 of those volatiles out. So why did we use a low temperature distillation? It's because exactly what you mentioned. We're worried about degradation. If we use a high temperature distillation, plus you get conversion of some monoterpenes to other compounds.
Oh, so yeah, I observed that.
Yeah, so that's why we selected the low temperature distillation process. It was gentle, and we were hoping then we would avoid degradation, but it's certainly not the concentration that's in the fresh berries.
Yeah, and also the experiment you did on cell culture, and you diluted 10 times for the volatiles, and I probably remember wrong, but I'm just wondering, from that range, do you have any ratio to, like, if human subject consume the berries, what's the amount should achieve as the cell culture's amount?
Yeah, another great question. One advantage of the volatiles, even though they're present at very low concentration compared to the phenolics, the limited bioavailability data, which has been done a lot just with individual monoterpenes, some of them are quite high, 10%, 15, 20%. That's a big advantage to anthocyanins that might be like 0.1%.
Yeah.
So those dilutions 10, what we use, 2, 8, 10-fold, 2, 4, and 8-fold for the volatiles. I don't know if we're there at the same concentration we'd be seeing in the plasma or urine. I think there's just got to be more work done.
Oh, I'm doing that.
Yeah.
I observed the phase 2 metabolites of the carnosic acid glucuronides and some other glucuronides as well. But due to I'm using LC, so it's hard to trap everything right now. But it's a good process.
Yeah. So do you know the bioavailability then of the,
not yet, but I will.
You're working on that. Yeah. No, that's good, because there's really more information needed in that area.
Yeah, it's really interesting and exciting of your presentation. I'm sorry you are retiring, but it's really exciting.
Well, I hope it works. I mean, you always have ideas as a professor. Most of them don't work, but I hope this one works. It'd be a good idea to go out, retire on, but it's, a lot of times the bioavailability is what shoots you down. You get great results from cell culture assays, so you really take those results with a grain of salt, because these compounds, you never know what you're going to get with metabolites.
Yeah. Thank you so much.
Yeah, you're welcome. Other questions?
Yeah, other questions. Ben, looks like you have a question in the chat. Did you want to unmute and ask, or do you want me to read it?
Yeah, sure. I'll go ahead and ask it. Thank you, Dr. Howard, for that presentation. What I was wondering is, I guess based on what we were talking about before with the degradation of some of these compounds, I was wondering if, there's been there's some studies showing that these phenolics, because they're such good antioxidants, they can degrade in cell culture. So I was wondering if you looked at that or you don't want to comment on that.
No, well, see, we did 24, that's a good question too, because I give students a hard time when they're giving cell culture results on the effects of anthocyanins, and they've incubated for 24 hours and they're seeing anti-inflammatory results and they say it's the anthocyanins. Well, the anthocyanins are very unstable at pH 7, right? Physiological pH. We know from when we used to run the ORAC assay for an hour, half of the anthocyanins had broken down into hydroxybenzoic acid derivatives after an hour. After 24 hours, there's not many native anthocyanins left. They're all the hydroxybenzoic acid derivatives. So that may not be good for the anthocyanins, but if you are seeing an anti-inflammatory effect, or it's the metabolites, the breakdown products of the anthocyanins, okay, that might be important. So no, I certainly wouldn't expect the anthocyanins to be stable in cell culture over 24 hours. You'd be looking at some of these hydroxybenzoic acid derivatives, fluoroglucinaldehyde, another one, the breakdown products of the anthocyanins.
Okay, great, thank you.
Sure.
And maybe to add on to that, we can detect those in plasma, right? The metabolites. Sure. So, I mean, it's a great point, and cell culture does get the criticism that you're describing because some of these things are just breaking down straight away, but some of them are breaking down to metabolites that we do in fact see that could be having the effect in vivo.
Right.
So, yeah, it's a really good point.
Well, and the same thing's going on too, of course, with the gut microbiota. I mean, the end result are a lot of these hydroxybenzoic acid derivatives, and hypuric acid is really high concentration in plasma and urine, probably higher concentration than the breakdown products of the natural polyphenols that are absorbed. Would that be right, Brett?
Yeah, I mean, it's really high. Yeah, you're in micromolar concentrations.
Yeah.
Sure.
So yeah, there's nothing wrong with the metabolites. If they're bioactive, that's great. May not be the native compound.
Yeah. Other questions for Dr. Howard?
No, I saw a chat. Somebody said hint is yummy. So yeah, if you haven't tried hint water, it's try it out. Just a berry flavor with no artificial sweeteners or anything. So yeah, natural flavored water.
Yeah, looking forward to it.
I do have
Indica or Aman, one of you two?
Yeah, I'll go first.
Okay.
Hello, Aman.
Hi, how are you?
Good.
Nice.
How are you?
I'm good too.
Good.
Great presentation. We
Thank you.
really enjoyed it since this is my area, so it's always exciting to learn something new. So I have a question. You did mention in your conclusion slide that there's going to be work on the synergistic action of the phenolic plus the volatile essence.
Right.
Did you do anything yet in your lab?
No, sure haven't. You're free to take that on because we need all this work done, and I'm pretty much done with it. So take a go at it.
I need a good student from your lab.
Yeah, you need money and a student. That's always the problem, right?
Yeah, no, it's great because the way we consume berries, like we consume them as it is. So right now there is a lot of data on the phenolic components imparting health benefits. But if this work is added on, I think there will be definitely some health aspect associated with the volatiles as well. So yeah, I look forward to that, like in future.
Well, I hope it pans out, but listening to a lot of the speakers at the Berry Health Benefits Symposium, including Dr. Burton Freeman, they'll talk about when you start looking at fractions, the phenolics, they might account for 60, 70, 80%, but where's the other, 20, 30% of the benefits coming from? And hopefully it'll get proven that it's volatiles, but only time will tell.
Yep.
And it could be other compounds too, like still beans or others that we haven't even looked at. There's so many different natural compounds in plants that
It's very
you know,
hard to track everything down. So
yeah,
it's like too complicated, I would say. Like one berry would be having so many different components, like apart from phenolics. So,
right.
yeah.
Because I mean, it could be compounds that are present at very low concentration too, that are just extremely bioactive. I was shocked when we took those standard volatiles, we took like the top 40 that were found in the blueberry at the in the blackberry at the concentrations found in the blackberry. We mixed those up. If you smelled it, I thought it would smell like a blackberry, and it smelled like a medicine kit, man. It wasn't even close. So again, it's these very potent odorants at very low concentration that give the blackberries their aroma. But then, so what does that mean in terms of bioactive properties? It could be these ones that are present at very low concentration. I mean, we don't have a clue to what specific volatiles are playing an anti-inflammatory response. You know, hypothesis was it'd probably be monoterpenes, but I mean, we don't know for sure. So there's a lot of work to do in this area.
Yep. Thank you so much.
You're welcome.
Super. Indica?
Hey Luke, how are you?
Good,
to see you.
Indica. Good. Good to see you.
Yeah. So my first question is, when you're planning to visit us?
Oh, well, I'll be in retirement. Oh, I
That's
always
right,
like
so-
coming up to the big city. It's
I
only
know.
about a 5-hour drive from
Yeah.
Bloomington. So yeah.
But we have great restaurants, so we can revise some of the things that we have been discussing before you retire.
That sounds great. Love to come
Yeah.
up to Chicago.
Yeah.
Hopefully
So-
catch a Cubs game too.
Right. Yes. So I have some comments, actually, not like question plus comments. So it's really good. I mean, fantastic experiments. This is what we have seen in our human models. So one of the issue that we have, we have when we give them our subjects, at-risk people, something strawberry, blueberry, or all the berries, we have seen that they are postprandial IL-6 levels and certain things significantly going down.
Right.
But one of the problem that we have is we isolate their monocytes, and then we are trying to understand the mechanism using NF-kappa B pathway.
Mm-hmm.
And we can see something, but we really cannot get that into the big picture. One of the reasons is that when you take the blood from your body, right, and the blood, now the cells are outside, and then the cells are getting stressed, and the NF-kappa B pathway activate very quickly.
Just due to the stress by taking it out.
Yeah. So, and then on the top of that, we try to isolate the cells using all the gradient centrifugation techniques, and those are putting stress. And by the time we go to the microscope, they are already all the NF-kappa B inside the nucleus. So we cannot see the changes.
Oh, wow. Yeah,
that's one of the things that we are facing now. One of the way to overcome this is use the flow cytometry, because you collect the blood and you can right away put in the flow cytometry, and then you can look at the separations, all the activation, deactivations.
right.
Because if you isolate the cells and if you go through that pathway, it is very hard because you are putting the cells under stress.
Yeah.
And that stress itself is translocating NF-kappa B subunit to the nucleus, and then you can see they are all activated.
Mm-hmm.
So then you cannot see the treatment related impact.
Yeah, they're already. I got you. They're already stressed out to the max just through.
Yeah.
Yeah. Well, man, that's the same problem. I mean, even when you purify any phytochemical from a plant, the more you purify, the more unstable it is.
Yeah.
So yeah, that's certainly a challenge.
Yeah. So
how
anyway,
to-
you have worked with those lot of compounds. Do you have those in your hand? We can just throw into our systems and see whether we can.
Like
some-
the volatile compounds?
Yes. Yes.
Yeah, we've got quite an arsenal of standards. That's the beauty. The volatile standards are actually pretty cheap.
Okay.
But the question is, intuitively, and we always do this, we try to pick the ones that are the highest concentration in the fruit. I don't know if those are the good ones or not for anti-inflammatory activity, but yeah,
Right.
I mean, if you wanted a handful of those things, we could certainly send them to you.
Yeah, I mean, if you can do something, then we can, I have some few things that we can talk offline, and I don't want to take other people's time.
Yeah.
Some experiment models that we can utilize kind of close to the in vivo system.
Right.
Rather than, completely in vitro, like cell culture.
Yeah.
We have some models that, little closer to the human body.
That's good.
Yeah.
Because actually, Laura, my PhD student that's working down at the Arkansas Children's Nutrition Center, she was taking some plasma samples from subjects that some had consumed blueberry products, and challenging those with LPS and looking at the volatile and I'd see a little bit of anti-inflammatory response, at least suppression of IL-6.
Yeah.
So getting closer to the in vivo model certainly is good.
Yeah. Yeah. We
so,
can.
I'd love to visit with you more about that.
Yeah.
Okay.
Yeah. Let's plan something.
Sounds good.
Okay, so I know that we're getting at the top of the hour. I do have a question in the chat. So I'll throw it out there. I know people may be falling off, and I thank everybody in the audience for joining us. So if you do have to leave, but we're going to keep, Dr. Howard around for at least maybe a couple more minutes. So the question in the chat is pretty long. It has to do with the solid food matrix versus liquid food matrix. And I don't know if you want to take a look at that.
Yeah, I'm looking at it now.
Might be easier for you to read and address.
Go to the phenolic change with a solid food matrix. Well, it's going to be, it's certainly bioavailability in a solid food matrix would be much different from liquid matrix. But we just looked at crude extracts from the berry but we did just get liquid extracts of the phenolics and the volatiles. In vivo, in the solid, it'd be different. But the challenge with the volatiles, and we wrestled with this, normally when you're looking at phenolics in cell culture, you extract them, with the solvent, you evaporate the solvent, you've got your solid phenolic extract that you can do a concentration gradient, right? Just diluting it. How would you do that with volatiles? You can't evaporate them. You've lost the volatiles. You probably could use supercritical fluid extraction, but you would also be extracting other compounds. So that's why we went with that low temperature hydrodistillation technique. But no, I certainly agree that the bioavailability would be different in the solid food matrix versus, an isolated liquid fraction matrix. Yeah, but that's somebody else got to do some work in that area.
Do you have a sense of how much may be absorbed through the respiratory tract? So through, they're sniffing, right? But then, you're smelling your fruit, but then there's also the retronasal.
Yeah.
So after chewing and mastication, so there's probably some percent that.
Well, there is. And the pulmonary, the papers that are out there, and with some of them, Brett, they've just used an individual monoterpene. And they show, I had that figure, and this one where I showed the one for linalool, I think it was 23%. But in a review article, it said it can be as high as 50 to 70% pulmonary.
Oh, okay, yeah.
So this is a good news for the wine drinkers, right? You smell the wine, you're getting all those volatiles from the grapes, and this is good news if that's right.
Right, we'll have to do more swirling and sipping.
I don't smell my beer that much, but maybe I should. But no, so in terms of, protecting against inflammation in the lung, hey, might be something to it.
Yeah, right. All right. I might have to bug you for a couple of those papers that you cited in your presentation.
Yeah, it sounds good.
Yeah.
I've got them, close by in the office. Although I won't be in the office much longer.
I know.
I'll get those to you soon. I'm still going to be coming in a little bit after I retire.
Super. All right. Well, thank you so much for your time and presentation today. This was really phenomenal, and really happy you joined us.
Well, thank you for the invitation. It's a great thing to do on a snowy day when the university's shut down. You don't want to listen to the news. It's like war out there. I mean, it's terrible news. So this is taken away from that, which is good.
Yes, indeed. All right. Well, happy Thursday, everybody. And for those of you that are still hanging around, we'll see you next Thursday. And Luke, we'll see you soon, I hope. Let's try to connect.
Hey, sounds good.
Yeah.
Thank you.
All right.
Thank you.
Yeah, stay warm out there. Good seeing you all.
Yeah, good seeing you too.
All right. Bye-bye.
Bye-bye.